In this video, we are going to be discussing the topics in Chapter 17, which has to do with glycolysis. We are now going to be looking at the process of metabolism, but we are going to be looking at the overall picture. As I expressed in the previous chapter, overall, when we are metabolizing through the process of respiration, a molecule of glucose, you can see that we're going to be reacting it with molecular oxygen, and the products are going to be carbon dioxide, water, and energy. That energy, as you can see, per mole of glucose is going to be negative 685.9 kilocalories. Now, this overall reaction, understand that it is a combustion reaction. But in biochemistry, this combustion reaction does not produce all the energy at once. What we are going to be looking at in this chapter is going to be the first stage in the metabolism process, because overall, to be producing that carbon dioxide, water, and energy, understand that in a way we have to think about it that we're going to go through 3 stages. And the first stage is going to be glycolysis. The second stage is the citric acid or tricarboxylic acid cycle. It is also known as the Krebs cycle. And then the third stage is going to be oxidative phosphorylation. Understand that stage 2 and stage 3 are going to come in later chapters. Now we are going to be going over section 17.1, which is the overall pathway for glycolysis. So understand that glycolysis is not as a whole the figure that we have in this slide. Glycolysis is just the steps that I have put in this red box. Glycolysis is only the process in which we take glucose. We convert 2 molecules of ADP into 2 molecules of ATP, and that glucose turns into 2 molecules of pyruvate. Okay? Now, understand, even though it's not outlined in the general reaction, in this process, we are also going to be generating 2 molecules of NADH. And we're going to see in later chapters how NADH is involved in the process of oxidative phosphorylation in order to make energy. Now, there are possible fates of pyruvates during or I should say after the process of glycolysis, and it all going to depend on the availability of oxygen in the system. So if we don't have oxygen, then the fate of those pyruvate molecules that were synthesized in the process of glycolysis is going to be either formation of lactate, which we have on the left side of the figure, or we're going to have alcoholic fermentation. And what we are going to be making is CO2 and ethanol. Understand that that second process is specifically found in yeast. If we have oxygen in the system, Then what's going to happen to pyruvate is that it's going to go through the citric acid cycle, followed by oxidative phosphorylation, and that's how we're going to be making that CO2, water, and energy that I expressed in the previous slide. Now, understand that whenever we don't have oxygen in the system, that is known as an anaerobic process. And when a process requires O2, that is known as an aerobic process. That's why here in the slides we have aerobic oxidation versus anaerobic. Okay? So let's look at the overall view of glycolysis. As I expressed previously, in the process of glycolysis, what we have is the conversion of glucose to 2-pyruvate molecules, and overall in this molecule we will be converting 2 ADP molecules that will be combined with inorganic phosphate to produce 2 ATPs. Remember, we also make 2 NADH plus molecules, and those NADH molecules, understand that they are going to be processed later. And just leave it as NADH. So those 2 NADH molecules are going to be processed later through oxidative phosphorylation. So I just want to make note that they are also produced. Overall, the process of glycolysis requires 10 steps, and they are each catalyzed by a different enzyme. Understand that the most common enzymes that we are going to be looking in these 10 steps are going to include kinases, isomerases, and dehydrogenases. For this section of the chapter, you guys need to be familiar with structure, the name of the enzymes, coenzymes that are utilized, and the products of the reactions. Now we are going to be discussing section 17.2, which includes what is going to be the first few steps that allow us the conversion of glucose to glyceraldehyde 3-phosphate. So now we are going to be going over the steps of glycolysis, again, the 10 steps one by one. Step 1 of glycolysis is the phosphorylation of glucose. And as you can see, this specifically is going to be requiring hexokinase as your enzyme. Hexokinase requires magnesium, which, as you can see, this is a divalent cation. ATP is going to be our source of phosphate. So when the phosphorylation happens of that glucose molecule at carbon 6, as you can see, the hydroxyl group is now phosphorylated on carbon 6. That phosphate group is going to come from ATP. So understand that this reaction, the phosphorylation of glucose, is an endergonic process, which means that it requires energy in order to happen. So where does that energy come from? Well, the hydrolysis of ATP, because it is an exergonic process, provides the energy in order for this phosphorylation to occur. Understand that this process is irreversible. Okay, so that's why we have the use of a single arrow going forward. This step is actually regulated by the product of the reaction. So glucose 6-phosphate actually inhibits the hexokinase enzyme to regulate this step of the reaction. So you may ask yourself, why is it that in the first step of glycolysis, we need to phosphorylate glucose? Remember, glucose enters the cell through facilitated transfer. So remember that in facilitated transfer, we're going to have a channel that is going to allow the glucose to come in. Now, why is it that we need to put a phosphate group on the glucose? Well, once we put the phosphate group, now we have a charged group on our glucose, and then that is going to prevent glucose to utilize that tunnel to get back out. So in order to retain that glucose inside the cell and not to put it outside the cell, the system decided to put a charged group on it. Now, overall, as you can see, the molecule is negative and it cannot utilize that channel in order to get out of the cell. So once you have this glucose charged with the phosphate, then you're keeping it in and you're taking it through the process of glycolysis. The next step in glycolysis is going to be isomerization. So, as you can see, the enzyme glucose phosphate isomerase takes our glucose 6-phosphate into fructose 6-phosphate. So what does this mean? That glucose 6-phosphate and fructose 6-phosphates are going to be isomers of each other. This isomerization reaction is through a keto-aldol isomerization. Understand that you guys don't need to know the arrow pushing mechanism of the conversion from glucose 6-phosphate to fructose 6-phosphate, but you do need to know that step 2 is an isomerization reaction. On step 3, we have another phosphorylation reaction. In this phosphorylation reaction, our enzyme is phosphofructokinase. As you can see, for this reaction to occur, we need a divalent cation. In this step, similar to what we saw in step 1, we are going to convert ATP to ADP. Step 3 in glycolysis is also an endergonic process. So this is a process that requires energy. Where does that energy come from? Well, from the hydrolysis of ATP. Remember that ATP is not only going to be giving the energy to make this reaction happen, but it also provides the phosphate group for the phosphorylation reaction. And the substrate that is being modified here by phosphofructokinase is the conversion of fructose 6-phosphate to fructose 1,6 bisphosphate. Understand that step 3, similar to step 1, is an irreversible step. That's why here we have a single arrow. This step, it is regulated specifically by ATP, but here ATP is going to be regulating phosphofructokinase allosterically. On step 4, we're going to take our fructose 1,6 bisphosphate and the enzyme aldolase is going to do a reverse aldol condensation reaction. You guys do not need to know the arrow pushing mechanism of the conversion of fructose 1,6 bisphosphate into the two products. You need to know the name of the reaction. Now, the aldolase enzyme will be converting the fructose 1,6 bisphosphate into two products, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Okay? Now, one of the things here is that, again, in the process of glycolysis, all these reactions are occurring. And you may ask yourself, why is it that in this step we created two distinct enzymes? Understand that the process of metabolism, we need to be talking to different pathways inside the cell. So dihydroxyacetone phosphate, we're going to see its involvement in other pathways. So glycolysis, it's in a way directing the utilization of glucose through a process that is going to result in making energy. But depending on the conditions of the cell, some of these intermediate molecules that are produced in the process, which are called metabolites, will be utilized in other pathways. So far in step 4, we are going to take a 6 carbon sugar, and we're going to be creating 2 3 carbon molecules. On step 5 of glycolysis, we're going to have an isomerization reaction in which dihydroxyacetone phosphate is going to be isomerized by the enzyme triose phosphate isomerase, and dihydroxyacetone phosphate will be converted into glyceraldehyde 3-phosphate. So understand that this isomerization is through a keto aldol isomerization. And by the end of step 5, we are going to have 2 molecules of glyceraldehyde 3-phosphate, because remember that in step 5, we're going to take one of the 2 products from step 4, aka the dihydroxyacetone phosphate, and we are going to be isomerizing it to another molecule of glyceraldehyde 3-phosphate. So understand that in the sequential steps of glycolysis, meaning steps 6 through 10, now, even though in the slides it may illustrate 1 molecule, indeed, we are actually processing 2 3 carbon molecules, okay, that came from glucose in those steps 6 through 10. So understand, steps 6 through 10 is actually 2 molecules of glyceraldehyde 3-phosphate that are going to be processed through those steps.